welcome everyone. I'm really, it's a pleasure for me today to introduce Emma, because the first paper I read on amyloid forming proteins, it was a paper from Emma when she was she wrote when she was doing her first postdoc in Utrecht. So Emma came from a biophysical chemistry field, and she got her PhD in 2001 in Sweden. Then she moved to Utrecht University in the Netherlands, where she performed her first postdoc. Then she did a second postdoc in Uppsala, and she was able then to start her own group in 2004 in Sweden, in Lund University. And she worked, and she's working still on amyloid forming proteins. Before she was working on IAPP and then alpha-synuclein. And one of her expertise, it's protein membrane interaction. She's working a lot with membrane and membrane models. And today she will speak about, I guess, alpha-synuclein and membrane and lipids interactions. So Emma, please.
Thank you. Let's start to share. And I hope it's now in the good view.
Yes.
Correct?
Yeah.
Okay, so thank you very much. And I will talk about decorated vesicles is the title of this talk. And it's focused not, I will hardly talk about amyloid formation, but the systems that possibly precede amyloid formation or not, because alpha-synuclein may also have healthy functions where that also important with lipid membrane interactions. So before starting, I start to show the main people who work together with me on this. So now there is, who can see this? Okay, there was a message, but that is not anything to care about. Okay, so alpha-synuclein is probably mainly studied because it's relevant, or because it forms aggregates that gives plaque in Parkinson's disease Lewy bodies. But it also is associated with other functions that are, it's believed to be associated with synaptic vesicle trafficking function. In both, so these are sort of both two different scenarios that also associate with two different self-assembly or co-assembly processes. And in both these cases, the interaction with lipid membrane are interesting. So to the left here, you see examples of what we have studied from a study quite some time ago, but that we have studied in our group for quite a bit, and that is the process of that when protein alpha-synuclein aggregates in the presence of lipid membranes, it aggregates, the aggregates that form can contain lipids, so we get basically lipid protein co-aggregation. So it's not only an aggregation at the membrane, but also lipids within the membrane, within the forming amyloid. In the other end, we look at example, and a more recent example, but where we studied the interaction of the monomeric protein and lipid membrane, where the yellow color indicates that we have both protein and lipids there. It's an overlay of green and red, and here we see phenomena that is not amyloid formation, but are rather membrane remodeling phenomena. And in both these scenarios, it is clear that the interaction between protein and lipid matters. And then one can ask oneself, I mean, when do we go one direction and when the other in these two scenarios? Here's an example of, and that is the only aggregation data that I would show. Here are data obtained from thioflavine T fluorescence of where we studied the aggregation of alpha-synuclein at different lipid protein ratios, so in the presence of vesicles at different amounts. And the plot give you the half time of the aggregation. So when adding vesicles to this, a little bit of vesicles to this protein, we can induce or speed up aggregation, but when we come to a large excessive vesicle, there is no detectable aggregation. So we can conclude that even if we only have the same components in the system, we can get completely different behavior. And there is somehow a tipping point between inducing aggregation and preventing aggregation. What we also can see is that in all these lipid protein ratio that we see aggregation, there is a free protein detected, as seen from this agarose gel in the solution, that is this bright band here. But in the other cases, there is no free protein. So in the conditions where we have no aggregation, there is no free protein. So to summarize this tipping point, at low lipid to protein ratio, we have bound protein and free protein. At high lipid protein ratio, we basically only detect bound protein. And in the cases where we have induced aggregation, what the free protein sees is not a membrane, it's a decorated membrane, it's a decorated vesicle. So it's the vesicle that is covered already with protein, which we believe is important. So the questions that I now will discuss are first, I wonder, how will this protein distribute and bind when it has sort of unlimited access to the membrane? Because that tells you something potentially about its preferred organization if there is an excess of membrane. And then we go ahead and look at the consequences of this binding, which could be, for example, membrane remodeling. I will then go on to see if there is we concluded that there is somehow a tipping point, aggregation or not, and what are the molecular differences or changes that occurs at this tipping point. Most of the work that if I don't say anything else, everything is done for the very same system. So we have alpha-synuclein and a lipid system that with a 30% charge anionic lipids, and this is sort of self-consistent. Everything is within this system, so that we can compare a lot of different techniques and basically only vary the lipid protein ratio. So here is somehow an overview of what I'm aiming to. So we already concluded that at a macroscopic or microscopic scale, we have amyloid formation only in the case where there's an excess protein. And on a molecular scale, we know that in the excess protein conditions, we have both free and bound, while in the other conditions where we have saturation or excess membrane, basically all protein is bound. I will start to focus on this side of this figure. And I wonder, how do the protein act or bind when it has access to an excess of membrane? From previous studies, it's well known since many years, it is known that the free protein is unstructured. When it binds to a membrane, it forms alpha helix, but with a C-terminal unstructured and pointing out. And I think you've all seen, if you read the alpha-synuclein literature data like this. So if one add alpha-synuclein, here is CD experiment to membrane, you see the formation of these alpha helices. And at some point to get to somehow an unchanged of the CD spectra that indicates a saturation point. And for the system that we study, it occurs slightly below lipid to protein ratio of 200. So now we add a protein to a membrane system where we have an excess of membrane. And then the first question is, so do we get sort of a random distribution of the protein, or does it go to a few vesicles or to some places at the membrane? And of course, from entropy point of view, the random case is the sort of expected case, but it doesn't have to be the case, and it is not the case. So this was a sort of a unexpected finding that we sort of stumbled on, but that we have then investigated in quite more detail. And this is how we sort of first realized. So here are giant unilamellar vesicles. Look at vesicles in the left picture here is in bright field, so we basically see all vesicles. Then add, the vesicles are not labeled with any dye. Then we add labeled protein, a small amount, so not to cover all vesicle, not below the, above the saturation point, so to say. With respect to membrane, we have a high lipid protein ratio. And then you see that some vesicles contain protein and some are bare. And this occurs at many, I mean, this is the observation that and adding more protein, more vesicle fill, and eventually all are filled. So the observation is that we actually have this situation of a cooperative binding. The protein goes to the place where there is already protein in favor for the bare membrane. To quantify this, we did fluorescent correlation and fluorescence cross-correlation spectroscopy, and then we used small vesicles. And what these data show you here, from the FCS data, we can count the number of vesicles that are labeled, in this case, either with a blue that is actually green dye, but it's plotted here as blue to make it easier for those that cannot distinguish blue and green. No, red and green. Yeah. And the red, so basically here we count the number of vesicles that carry the blue dye, say, and we keep the amount of protein constant and add vesicles. And of course, if we add vesicles, we see more vesicles. So that's natural, these blue dots. The red dots indicates the number of vesicles that are both green and red, or blue and red. And then we see that up to a certain point, that it goes with the number of vesicles, but then it levels off. So this means that at high lipid to protein ratio, we basically have both free vesicles and vesicles that have both green and red dye, the cover protein. So there's a coexistence of bare vesicles and filled vesicles. And the filled vesicles are then, if you compare to this saturation point, it's pretty closely packed with protein. Also in a membrane-free system or a dye-free system, we can sort of get support for the same. So here it is so, and I will come back to this in a little bit, but it's also since a long time an observation in the literature that binding of alpha-synuclein to a vesicle actually lead to vesicle deformation. So we get this prolate shape of the vesicles that you see in these cryo-TEM images. And in these images, the lipid to protein ratio has been varied. And as you can see, if you look in more detail, the fraction of vesicles in these images that are deformed actually vary with the lipid protein ratio. And in this, where you have a lipid very large excess of protein, of lipid vesicles, you only see few vesicles deformed. So this again supports we can quantify it here. Let's see there. So that when you come above the saturation pressure, you basically start to see that the proportion of the deformed vesicle decrease when you increase the lipid protein ratio. And that sort of supports this conclusion that I presented before, that if you basically have two populations, bare and covered, decorated vesicles, and when you go to large excess of vesicles, of course, you get more of the bare ones. So the conclusion for this is that we have the strong cooperativity in the binding of alpha-synuclein to the membrane. So this means that the affinity of the protein to the membrane is much higher to a place where it's sitting beside another bound protein compared to a bare membrane, which is completely against what would be predicted from entropy. So it actually also tells you something about the interaction of the membrane, that it has to be very highly favorable to go to the same place. And as a consequence of this, you can, at the situation where you have a very low amount of protein in your system and an excess of membrane, still have a locally very high concentration because it goes to the same place, basically. Then what are the open questions after this are, of course, maybe the second one among these open should be the first one. What are the underlying mechanisms? This is, at this point, unknown, and we have ideas, of course, and happy to discuss. And one could also wonder what are the consequences of this? Does it have any effects? And potentially one can also discuss whether this actually is something important for the healthy function of the protein. But I will now talk a little bit on the effect of lipid remodeling. So I showed you before and this, that the binding of alpha-synuclein to the membrane has an effect of deforming the vesicle. So here's a cryo-TEM image of just a pure vesicle suspension, and you get spherical vesicles as expected. Adding alpha-synuclein, and this is just freeze just after the addition of protein, you clearly see deformed vesicles, and the degree of deformation depends on the lipid to protein ratio. But typically we get this prolate shape. Another observation is, as you see in this image here, is that basically you get no change in shape if you wait 24 hours. So previous studies have suggested that alpha-synuclein induced vesicle fusion. That we have published papers. And then these kind of structures that are seen here with this little neck and so on are interpreted as fusion intermediates. For us, this was led us to think we were a bit confused about this finding. So have tried to look into it a little bit. Because of course, when you see this kind of structure, it could be a fusion intermediate, but it could also be a fission intermediate, or it could be somehow a stable deformation, which appeared to be the case for this lipid system that we study here, as we actually get no change after 24 hours, which would, as bending a membrane involves an energy cost, one wouldn't expect the fusion intermediate to remain over 24 hours. Also, we see it is, the binding, or at least asymmetric binding, as I will come back to later, of alpha-synuclein to the membrane promotes structure with more positive curvature. That means that you go to a smaller object, and that could potentially be a reason driving towards fission, but it's hard to see together with fusion. So with this as a background, we decided to look a little bit more into this. With the first question, is what we see here fusion, fission, or stable deformation? Fusion, fission, intermediate to stable deformation. And again, we did fluorescence cross correlation. And here we had no dye on the protein, but we have green vesicles. They are blue here, yeah, for the same reason as before. Red vesicles. And then we both record the fluorescence correlation, autocorrelation function for one type of vesicles and the other. They are not scaled to each other because the background is different. So this is just the raw data. And the black line here is the cross correlation. So that means signal from those vesicles that contain both green and red dye. And in this case, where we've just mixed the two vesicles, you will at sort of reasonable time scale have no mixing of them because the lipid solubility is low. And therefore you see cross correlation that has no intensity. So there is no co-localization. Then we add protein and we more or less add the same. So this is at the lipid protein ratio 50, and at even higher concentration, there is no cross correlation. So that no cross correlation means that there is no vesicles that contain green or red dye to any detectable level, meaning that there is no detectable fusion. We can also, by analyzing these data and taking into account the background signal, extract information from the intensities and the background correction, the number of vesicles over the different lipid protein ratio. And here it is more or less constant, as you see here, which means that there are no signs of having fewer or more vesicles. So basically no detectable signs of fusion or fission in this case. So the conclusion from that case with this 30% lipid, charged lipid, is that we more or less have stable deformation. But of course, if we change the system, maybe we can drive it even further. And one question that one could, of course, ask is that what the binding of the, I said that we have the protein, when you're vesicle, the protein can basically only access the outer side, and that is what we believe is what's leading to preferred highly positive curvature. What would then happen if we have a symmetric binding of the protein? And we can look in this to the left, upper left here, you have symmetric binding of the protein. You see these pro-like structures. In the middle, you have no protein at all. And on the right, you have symmetric binding of protein. So the protein is added before vesicles are formed. And these basically are identical to the they are spherical, so not deformed. And in the lower side, you see that they also respond to external pressure in a similar way as the asymmetric, as the bare membrane. So basically, asymmetric binding is a key, and that's not unexpected, of course. Then we change the lipid composition, and we look at the distribution of vesicle size, because if we had a fusion or fission phenomena here, one could expect, of course, that the size distribution would change. And now we, instead of changing the way the protein bind, we change the lipid composition. And here we have lipid with the same system to the left as we had in all other examples. The middle figure is when we have ganglioside, which is a lipid with actually by itself from micelle has a huge head group, so it's a cone-shaped lipid. And then in the right figure, a very popular model system that contain PE, which is a sort of have a small head group, so it promote reverse structures. And by analyzing the size distribution of vesicles, we can see that first, in none of these cases, we get any objects that are bigger, so there are no signs of fusion, but potentially there are vesicles that are smaller in some of these cases. And of course, here we have a not very, in a way, good method, because we have a 2D image and a that we analyzed with respect to 3D objects. And a prolate looked from the long axis would look like a small vesicle. So one should probably do something else if one want to understand if we could drive fission in these systems. Therefore, we did single fluorescence microscopy, where we again had this setup where we have vesicles with two different dyes, and we mix them. If we have fusion, we would expect to get vesicles with mixed colors. Also, if there is an exchange, that would happen. And if we have fission, we will simply get more vesicles. And what we can do in these experiments is again to look at co-localization, and we see no co-localization, so conclude no fusion. And we can also see fission. We can also count the number of fluorescent objects, and then that could indicate, for example, if there is fission. Here is data, and you can look at the left figure. So here we have compared two different systems. And this is the change, delta N here, is the change in the number of vesicle at two different time points compared to the case where there is no protein present. If we have the standard vesicle, so to say, that I talked about before, we see hardly any effect on the number of vesicles, so that there is no major change, meaning there is not a major remodeling, probably not much fission, but inducing gangliosides in the membrane, which is a common lipid in neural membrane, we actually get a very high effect on the very strong behavior that we get a spontaneous, we get much more objects, so that indicates a spontaneous fission. So to conclude, we can say that the asymmetric binding of alpha-synuclein promotes higher curvature, that the protein binding leads to stable vesicle deformation, and that when we have gangliosides there, there is spontaneous fission, and there is no detectable signs of fusion. So here I'm now trying to fill in here on this side. So we basically see that we have the cooperative binding I indicated here. The light color is that we have basically no protein free, and that we have protein bound with a full segment. Now we take a little, the 5 last minute look on this side. So there we know that the protein, we have a lot of excess protein, and the excess protein see these decorated vesicles. So how do they look like? And I only shown you this, on the lower side, is with a full alpha helicals bound here. If you look in literature, this is an old figure I showed you in the beginning as well, but I just bring it up again. So here are typical data for alpha helix formation when we titrate in lipid membrane. And we come to a point that we get an increased alpha helix content to a saturation point. But of course, CD is a measurement of the amount of alpha helix. And that could be to certain, I mean, a certain CD signal can be from a large alpha helical segment in one protein or to a small in many protein. And therefore, from this kind of data alone, we cannot say whether the same segment is bound to the membrane for all. I mean, is it always the same way? Do we always have this 100 amino acid bound? And as I will show you soon, that is not the case, apparently. So for this, we did HSQC NMR on 15N labeled alpha-synuclein. And from this, we can basically detect signal from the segments that are freely moving. So, and when then here is typically the different residues of the protein, and we have high signal everywhere. That is the protein in solution. Now we introduce vesicles in the system. And what we then see is that the signal decrease for not for the C-terminal part, but for the 100 first residues of the protein. But if you look a little bit in more detail here, you see that for the lipid protein ratio of 100 and 250 and 200, that is the yellow and purple, you basically lose all signal from segment 1 to 100. And that is a sign that this is I mean, you basically have what I showed in this picture before. The 1 to 100 segments are bound to the membrane. But when you come to a situation where basically you are below the saturation point, so there is a limited access to membrane. There is less membrane than what the protein would like to have, basically, if it would be all of it bound. Then you start to see binding modes that includes, that you see signal here means that from 60 to 140, the a fraction of this part of the protein is not bound. So this means that you have different bounding modes. In all cases, all of the first 20 amino acids are bound. It's like you're having a little feet in the membrane and the large part sticking out. While you can also have the binding mode that 100 amino acids are bound to the membrane and only 40 sticking out. So and I have not time to go through this in detail, but you can analyze this and say that there is a coexistence of these different binding modes depending on the lipid protein ratio. From this, we then can say that it doesn't look the same in all these conditions. The surface doesn't look the same. Further analysis of this also indicate or also show that the dynamics, the exchange rate of these different binding modes is not the same. And an implication of this is, of course, that this CD data measure of alpha helix signal is not directly proportional to the amount of bound protein. So what does the free protein see? Yeah, in condition where amyloid aggregation occurs, the free protein, it see the decorated vesicles where the protein is present in both bound and fully bound and partly bound state, which is very different from the situation that we have when we reach saturation. And this then builds up a figure where we see a clear difference both on molecular scale and colloidal scale and microscopic scale at this tipping point. And I skip this. So to summarize, we see a strong cooperative binding and a dense layer of protein at the membrane, also at low protein concentration, which induce vesicle deformation and for some conditions also fission. Then the decorated protein may induce amyloid formation, but that is not the same vesicles as we see in these excess membrane conditions, but a vesicle that have also protein bound with smaller fragments and that sort of are less stable interface, that have a faster exchange with the solution. And that was my summary. Thank you.
Thank you very much, Emma. So please type now your question in the Q&A down, and I will read it.
There are no questions.
Not yet.
I have a brief question for you, Emma, because you very quickly mentioned the effect of ionic strength. Can you give us some more details on this?
Yeah, so we have, yes. So first of all, one observation that is not only done by us, but also others, is that if you increase the ionic strength, I mean, normally you believe that that is something that, because you reduce the, or you have, you screen electrostatic interactions, and then you would potentially expect that you promote aggregation, but actually in this system, you prevent aggregation by increasing ionic strength, so adding salt, which one can explain by that the protein, the vesicle surface is mainly negatively charged, but the protein has a rather, I mean, it contains both negative and positive amino acids, and it has somehow a positive patch that, so for the attraction to the membrane to occur, you have an electrostatic attraction that you screen by adding salt. So that is one effect of increasing ionic strength, is that you actually make the aggregation slower. Another effect is that the saturation point comes at a higher lipid protein ratio, but we can still see, at least to some level of salt, this cooperative behavior.
Okay, so thank you. Now we have some questions. So the first one is from Jan Fischou, and he asks, how much does your saturation concentration and binding modes depend on the lipid composition of the SUVs?
Okay, good question. So the binding modes, if you have an excess of membrane, as far as I understand, I mean, all system we have looked at, the binding that you bind with this 100 amino acid is the same. If you change, for example, the content of charged lipids, the saturation point will move. At least if you go to, I mean, if you have 5% or 10% charged lipids, you have a saturation point that is slightly higher. I think that if you increase it above 30%, as we had here, there is basically no change. So it is, of course, a balance between, I mean, as there is an attractive component, if you have a low charge density at the surface, the affinity to the protein, I mean, then you that is lower, but at a certain point, it doesn't matter if you increase the charge because the surface is already closely packed.
Okay, so the next question is from Olga Gursky. She said, very nice work. Could you please comment on the effect of lipid charge on synuclein binding on vesicles remodeling?
So I think I sort of did it already. You mean on the vesicle charge? Yes. Yeah, so you have an effect of vesicle charge up to a certain content of charged lipids. So you basically get qualitatively the same behavior, but the saturation point comes at a higher lipid to protein ratio if you reduce the content of charged lipids.
Okay. The next one is from Tom Rothstein, and he asks, do these things, especially inhibition of aggregation, occur with natural extracellular vesicles, such as exosomes, derived from mammalian cells that already have protein in the membranes?
The inhibition, I mean, the prequistic for it, I mean, if you look at it somehow, I would say that the inhibition occurs in the condition where you basically take all the protein from the solution and bind it to the membrane. So you deplete the solution from free protein. That's the condition where you have inhibition. If you have membranes that are already covered with protein, I would clearly expect that that then corresponds to a situation where you sort of move to the left in this schematic illustration. So you basically need to count the protein that is already there. But so I believe it's the important thing is not the lipid content of these membranes, but it is the ratio, total ratio. So it has to include the membrane that you mentioned in your question. The protein that is already there has to be counted. Because you can basically only drain the solution from the protein if you have membrane surface to cover. Was this answer clear?
Well, I hope. If it's not, Tom will probably write something else after that. But then we have some question from, I don't know who, an anonymous spectator. So, did you compare the structure of the fibrils depending on the lipid ratio?
No, we have not. Well, we have which I didn't show you at all here, but we have studied co-assembled or co-aggregates formed for different types of lipids, and morphology-wise they are different, and also the uptake of lipid can depend on, for example, the charge of the lipid or the charge content of the vesicles. If you add an excess of vesicle, you will have fibrils decorated with small vesicles. So you basically to a certain level, it's in, one see aggregates where you can confirm the presence of lipids inside the aggregates, but no intact vesicles. If you further increase the vesicle content, which is I think what is asked for in this question, you see these aggregate, but also vesicles decorating the surface. So that's an effect of changing lipid to protein ratio. But these are all basic in conditions where you have an excess of protein, where you have aggregation.
We have a question from Gunilla Westermark, and she says, interesting talk, Emma. What about carbohydrates?
I don't know.
I will jump to the next question from Kathy De Wyson, and she said, great talk. If I understand correctly, your data shows that fusion of the liposomes does not occur at higher lipid protein ratios. How do you explain the increase in size of the liposome under this condition if it's not fusion?
But we don't see, we never see an increase in size, ever. We see potentially a decrease in size for some of the systems.
Okay, and we have Hams wants something. He said, do you think the observed NMR signal are only from free synuclein population, or do you expect contributions due to the exchange between free and membrane-bound populations?
I clearly expect, sorry.
Yes. If so, what about the conformational change between free so random coil, and bound, which is helical populations?
For the first part of the question, yes, I expect there is a contribution. So we see not the membrane associated part, which is here the alpha helical part, right? We see signal from the alpha-synuclein that is in solution, and as you saw also from this C-terminal part of the alpha-synuclein that is in the protein that is bound but is sticking out. So there is a contribution from that, but there is also a contribution from the fast exchange, from the exchange between. Do you expect contribution to the exchange between free and bound? Yes, for sure. We are analyzing this. That's my next talk. Yeah.
So it will be your next talk in Bordeaux in September.
Yeah, then I will talk about that. So we are currently analyzing the exchange time of the different binding modes.
Okay.
Which is different.
Thank you.
So the fully bound and the short and the bound with the little foot have different exchange time, and from a combination of NMR experiments we can get something out of that. But I think it's a little bit too early right now. I will tell in Bordeaux.
Yeah. So we have a last question from Takahiro Muraoka from Tokyo, and he says, thank you for the great talk. In the deformed vesicles, does synuclein bind at both the curve and extend areas, or synuclein bind selectively at the extended area?
So we have made quite a bit of sort of attempts to try to figure this out. It is, I mean, it's not so easy to know from these cryo images because you don't see the protein. We tried with some gold nanoparticles, but then they start to change the system. But if you simply do this thing that you make a calculation of the area of the bound of the 100 amino acid helix at the surface, if it how much space that would take, and compare it to the membrane formed at that saturation around 200 lipid protein ratio, it wouldn't be enough surface to only be on the highly curved tips. I believe it's more or less covering the vesicles, but probably with uneven distribution. But it's not an you have more. You have too little membrane surface to only make use of the tips, so to say, of the vesicles, the highly curved side.
Okay, so there is no more questions. So I would like to thank Emma again. And now I will let Robert Griffin presenting our next speakers, Loren Andreas. So I will.
Sorry. Okay, so it's my pleasure to introduce Loren Andreas this morning. Loren was an undergraduate at Oberlin College, where he started doing NMR with Manish Mehta. He graduated in 2007 and came to MIT as a graduate student at that point and joined our research group. And during his graduate career, he did many interesting things. Probably the most notable was that he determined the structure of the influenza A dimer of dimers and lipid bilayers. It's an M2 18-60 construct. And it was one of the very early structures of this particular molecule. And it was very good. It was a very nice study because it showed people how to do structures of membrane proteins and lipid bilayers. During that time, he started doing proton-detected experiments in a collaboration we had with Guido Pintacuda and Lesage and Lyndon Emsley at the ENS Économel in Lyon, France. So he was traveling back and forth doing experiments, and then decided that it was such a great experience that he went there as a postdoc from like 2014 to 2016. And during that time, he developed some of the very first ultra-fast H1 detection experiments, proton detection experiments, which today are more or less a routine tool to study both membrane proteins, amyloid proteins, and other systems as well. And in 2016, I learned from Christian Griesender that he had appointed Loren as a group leader at the MPI in Göttingen. And in 2018, he received an Immunutur fellowship, which I think he currently still has. And so since 2016, he's been a group leader and Immunutur fellow at the MPI in Göttingen. Now, Loren has done many interesting things, and as a consequence, he has won several awards, for example, a Marie Curie Fellowship. He was an ISMAR, International Society of Magnetic Resonance Young Investigator. And in 2019, he was the CNE News, one of the CNE News Talented 12 Young Investigators. And today, he's going to continue the discussion of alpha-synuclein and lipid interactions, I believe. I'm not sure exactly what he's going to talk about, probably binding of alpha-synuclein to membranes studied by NMR. And maybe he will tell us about the interaction of the favorite drug of the Göttingen group. I think it's ANLE 138B binding to synuclein and maybe some other amyloid systems as well. So Loren, the floor is yours.
Thanks so much, Bob, for the kind introduction. Let me share my screen. Just a moment. Okay, there it goes. I don't know why it's so slow today. So do you see the slides?
Yes, we can see
Okay,
it.
perfect.
Yeah.
Yeah, so thanks again, Bob. I will not actually talk about the small molecule at this time because actually Leif and Christian both covered a lot of what we had done with that small molecule in the seminar series last year or so. So I'll talk about the structure of a species that we've been able to isolate of alpha-synuclein, indeed through a procedure involving lipid membranes, vesicles. Before I start, I want to just say that this is a really collaborative effort among many people, both from here and other places, which I'll show in the acknowledgement slide at the end. But just to point out that the work I'll show you is really work from Vrinda Sant, both at the very end of her PhD and now as a postdoc here in Göttingen. So for this audience, I think alpha-synuclein really needs basically no introduction, and in particular for everyone who was here for the first seminar of this session. You know then that alpha-synuclein, at least in the first 100 residues or so, binds to vesicles as an alpha helix. And then if things go right, this is the way you see alpha-synuclein. If things go wrong, the protein can aggregate either into somewhat smaller species, which we call oligomers, or larger oligomers with a high degree of order. These are amyloid fibrils, of course. And these can spread throughout the brain and cause a variety of pathologies. Here's a picture of Lewy bodies with fibrillar alpha-synuclein. And these show up in Parkinson's disease and related disease, such as dementia with Lewy body or multiple system atrophy. So looking in more detail, you see kind of cartoon representation of these first 100 residues or so forming a helix when they interact with vesicles, and in particular negatively charged vesicles, as we just saw. The oligomeric species of synuclein are particularly difficult to study because of several problems of isolation and size. But a paper in 2017 began to characterize one particular species which showed interactions with membranes, and a hypothesis has been put forward that these may be toxic due to disruption of membranes, possibly forming pores. And in support of that idea, the fibril species are also seen to colocalize with lipids in here shown in Parkinson's disease Lewy bodies. And so we're really interested in characterizing these types of small oligomeric species. So let me introduce to you the way we prepare these samples, starting from alpha-synuclein dissolved in solution. And this work really goes back to Leif Antonschmidt, who was a PhD here until recently. And he carefully developed this protocol for generating and isolating and small amounts, but a oligomeric species of alpha-synuclein. So we start with a mixture of POPC and POPA under ultrasonication conditions, which generates small unilamellar vesicles. And then over time, we see the development through THT fluorescence of fibril species. The time scale on the horizontal axis is quite variable from batch to batch. And so this figure is scaled in terms of T over T lag. The lag time taken as an extrapolation of the slope in the middle here. And we see then by NMR chemical shifts that there's a progression of this folding onto the membrane and aggregation in the presence of membranes, which ends, of course, in the fibrils, but also proceeds through what we call intermediate 1 and 2, which are distinct in chemical shifts in NMR. So I'll first focus on the fibrils, in part because anyone who was here last week saw Gunnar Schroeder's talk and has already seen the fibril species, which can be structurally characterized in great detail with cryo-EM. So this is published in Freak et al. 2022. From the NMR chemical shifts, we can characterize the secondary structure, and we see that there are 5 beta sheet elements and highly ordered as would be expected for fibril species. So in cryo-EM, to our surprise, we saw some very interesting species which had never been seen before, and this is probably the most interesting one, where we see about a 20 angstrom separation in the protein density, and the only thing that can really be filled in between is then lipids. So lipids are really the glue that holds together these different filaments in this fibril structure. This shows just the L2A structure, and Gunnar's talk goes into much more detail on some of the other species that could be determined with cryo-EM. Based on the NMR, we see a homogeneous set of chemical shifts. So the basic element is predominantly the same and homogeneous in the NMR samples, although the quaternary interactions across the filament may be different. And then we were also interested in characterizing these lipids in more detail. And so in MD simulations, we see that these lipids do stably form micellar kind of like structures in between the filaments of fibril. So this can then explain these more fuzzy densities that are seen in between the fibril species. NMR spectra also back this up. So taking a look at the phosphorus NMR spectrum in a static sample, we see that while vesicles show the characteristic powder pattern, we see just an isotropic shift in the case of the final fibril samples, indicating that we have high curvature and consistent with these micellar type of structures that are seen in the MD. So this covers the fibril. Then looking more detail at the segmental folding, which occurs during this process, we see for the general case, there's a loss of helical valence signals, which are overlapped in monomer, shown here in green in the first panel. These disappear, indicating that the helix bound to the membrane is lost and has gone into other structures. We see valence 74 show up quite early in intermediate 1. So this tells us something about this segment of the protein forming a fibril-like structure already in intermediate 1. On the other hand, if we look at S87, this shows up very late in the fibril. And if we look at threonine 54 or 64, these only show the fibril-like chemical shifts in intermediate 2, but not in intermediate 1. So partly motivate the interest in this intermediate 1 is the connection to, let's see, sorry. The connection to toxicity, and we're not the first to notice that small oligomeric species of alpha-synuclein can have higher toxicity than fibrils, and this is also the case for this particular preparation, where you see that intermediate 1 shows the least cell viability in neuroblastoma cells, whereas fibrils really aren't particularly toxic in this assay. They have comparable toxicity to a bit higher concentration, 0.6 micromolar monomer. So this intermediate 1 species appears to also be more toxic than the fibrils. So in the following few slides, we'll do a little bit more, I'll show a little bit more comparison of the intermediate 1 and fibrils chemical shifts. So we see, looking at similarity, the green colored regions show basically the same C alpha and C beta and CO chemical shifts. These include a part of the very end terminus, which is still helical, as well as one of these beta sheets and a turn. And then in yellow, regions which also adopt very similar structures to the fibril, which have C alpha and C beta shifts still in the same regions. And then in red are the regions which are most different, and these include the C-terminal stretches, which in the intermediate have not yet folded to beta sheet secondary structure, but also beta 2 and beta 3 region of the fibril, which in the intermediate also shows beta sheets, but very different chemical shifts. So the beta sheet core has already formed in intermediate 1, this interaction between beta sheet 1 and 4, which you can see here in the schematic. This is already formed, and probably also the turnaround residue 59 or so has formed. But beta sheet 2 and beta sheet 3 appear quite different in the fibril. So how could this be? Well, one of the things that came to mind fairly early was whether beta sheet 2 and beta sheet 3 could be in an anti-parallel alignment arrangement. And we initially recorded proton-detected spectra, HNH and HCH in 3 dimensions to try to address this question. The NH spectrum of this protein is actually quite good. We have technical problems to deuterate, so this shows a protein without any deuteration. And if we spin even at 55 kilohertz, but go to high magnetic field, the resolution is actually quite good, good enough to start doing these atom resolution structures, or getting atom resolution structural data. And with these initial HNH and HCH spectra, we were able to identify a couple of contacts which are clearly in violation with the fibril structure, as expected. These are quite different structures here. However, on one side, identification of a contact relies just on a single proton chemical shift, which is then difficult to find unambiguous cross peaks, since there's quite a lot of overlap in the proton spectrum. So we can find some isolated side chain peaks for example, in the amine region, some methyl groups which are more shifted, and these can identify a couple of contacts. But this really didn't answer the question whether this is now a anti-parallel beta sheet structure. So to do that, we need to improve the method, and this is something that we do here in Göttingen as well, is to continually try to improve the methods. And just to show the problem a little bit more generally, when we do broadband proton recoupling, really the signal is quickly diluted into a large number of protons. On the right, you can see that even within 5 angstroms, there's 25 protons. So if we do a mixing scheme which recouples a proton to all other protons, then the signal is quickly diluted. And this problem is even worse because the amide-amide contacts that we might be most interested in are actually a little bit longer distances than many of the distances within a residue, which are 2.2 2.4 angstroms, and across the beta strand. In an anti-parallel stretch, 3.3, 3.8, and in a parallel stretch, even slightly longer. So the side chain distances then dominate, and most of the signal goes there and is lost. So what we need is a selective recoupling sequence, so we can only excite correlations between, for example, these amide groups. And this is what we did with a pulse sequence, which we call MODIST. And I won't go into the detail, but just show you how this looks for a very ideal system, microcrystals of SH3, and now showing the nitrogen-nitrogen dimensions that we can frequency encode indirectly. But this is proton-proton mixing. You see many, many correlations, almost as many as can be done with a deuterated system. So with this method in hand, we then turn towards the intermediate again. And here, let me start by pointing out the black cross peaks. These come from the beta strands 1 and 4 region, which we saw were in, based on chemical shifts, were in parallel and register arrangement, just like the fibril. And indeed, we see primarily sequential contacts showing up in these black colored peaks. And then in green, I'm showing you the beta strands 2 and 4. I've renumbered the index to start from the beginning of that anti-parallel segment, and we see characteristic cross peaks between 0.14, 2, and 12. So this is consistent with anti-parallel arrangement, and then also between 3 and 11 and 5 and 9. Now these distances are also quite far in the fibril, between 9 and 11 angstroms, and clearly inconsistent then with a parallel and register type of arrangement. The next question that we had was which of these are an intramolecular interaction and which are an intermolecular interaction. And for that, we used a 50% diluted sample, which has 1-to-1 unlabeled and labeled alpha-synuclein. So an intra type of contact should show us full intensity compared to the diagonal, whereas an intermolecular cross peak should show us half of the signal compared to the diagonal. So with this, we could identify which of the two is intramolecular, which is intermolecular, and build up then a basic dimeric unit, which is shown here. So we schematically then have beta strands 1 and 4 in a parallel and register arrangement, and 2 and 3 then in anti-parallel arrangement. The next logical question is to ask, how big is this intermediate? What is its oligomer number? Looking with negative stain cryo-EM, negative stain for the moment, EM, we see quite small particles. Compared with fibrils on the right, you can see some change in the contrast here, but really very little. And our efforts with cryo-EM of the intermediate one so far did not yield even a low resolution map. So this has not yielded something yet. So we turn to other methods, and one of these is to apply chemical cross-linking. And there we saw under several conditions. So first of all, the monomer with lipid around did not show anything except for monomer in the gel after cross-linking with DSS. On the other hand, the intermediate one does show something in the range of dimer, and maybe a hint of something towards tetramer. But this was not particularly conclusive evidence. This arrow just then highlights the dimeric species, which was fairly strong when the system was stabilized with 70 micromolar monomer. Okay, those are tetramer peaks. So the next thing we tried was to use an NMR method called Codex. This is something that Mei Hong has used before to determine the oligomer number. And essentially, our starting signal on the left will decrease to 1/4 if we have tetramers. And we do see a decrease roughly to 1/4. Compared to the fibril loses intensity faster, meaning that the distances between, in this case, labeled histidine are somewhat closer. However, you see that even the fibril only decays down a little bit below 5 mer. And so into this higher range of oligomer number, the method is getting somewhat less precise. There's also considerable background in this in the aromatic region, and this is probably what leads to some of the noisy looking data for the single carbon labeled histidine 50 sample, which we used for this codex data. So at this point, we could tell, even in light of these kind of issues, we could tell that the sample was at least a former. But of course, if there were assemblies that have larger histidine distances than this method can detect, then we would also not be able to detect them. So the next thing we tried was through collaboration with Wahid Songdakhtar and Hisham Mazal at the NPI for Light, is to attach a Atto 647 dye in the C terminus of alpha-synuclein, where we were lucky enough that this does not interfere with the aggregation kinetics too much. So the data that I'll show you has 1:4 ratio, a 1:4 ratio between unlabeled and labeled and dye-labeled alpha-synuclein. We also have some other data with 1:1 labeled. And with this, and together with 2 different methods which they use, we are able to access the oligomer number. So the first way of doing this is to look at bleaching of the dye and to see how many bleaching steps occur. So in the example that you see here for the bleaching data, you see 2 steps occur, and it's possible to count how many steps occur and then thereby infer the number of labeled dyes in that particular oligomer. And the second is to use their method called cold. First, I'll just show you an overview of how some of this data looks. So this is data acquired on a cold microscope at 300 Kelvin after spin coating in PVA and vacuum drying. And you see here in a 80 by 80 micrometer field of view, the different light sources for the fluorescence. So the second method is called cold, and this relies on the fact that different dyes have different orientations, and then the polarization of light that's recovered can be then used to distinguish one dye from another, even though they're very close together. And so after signal averaging, it's possible then to locate different dyes in a 2-dimensional plane, and this shows one such case where there are 4 different locations of dye detected. And then by doing some statistics on the probability, you see that as expected for only 25% labeling, most of the time only a single dye is seen, but there's also a population of 2, 3, and 4 dyes detected. So with this, we were quite happy to see that the number 4 for the oligomer actually agrees with the earlier data, both the cross-linking and the codex NMR data. The right, I'm just showing you a histogram of distances that were detected from the cold data. And I just want to say here that since we've labeled with the dye in a very flexible part of alpha-synuclein after about 40 residues, which are expected to be flexible, that C terminus there, that we see a very wide distribution of distances consistent with that high degree of flexibility. So while some of their other work is possible to then reconstruct a 3D image, since we have a high degree of flexibility here, that's probably not possible with this dye location. So with the tetramer in hand, then we can start to build some models of a oligomer, including 4 copies of alpha-synuclein. And I want to say up to this point, with the NMR, we have atom resolution information about the structure. So we really have high resolution structural information for a local segment, but since NMR is a very local method, the higher order structure is something we have to look at through modeling. And so this shows you a partial barrel type of model where it has open ends. The parallel and register segment is colored here in the green and purple, the antibarrel segments in red and yellow. And then we can also model some barrel type structures where we close this interaction and the interbarrel section then forms a barrel-like structure. These are, of course, very appealing looking, but we don't have any direct evidence for this type of structure at this point. We do see only a single set of chemical shifts, and so that's one of the things we have to consider when looking at these. But what we do also learn from these modelings is that both of these can form structures with quite a low target function, 7 and 9 for these two in cyana, and target functions below 10 really don't have serious clashes in the structure. So, okay, in the last few minutes, then I'll show you some data we have on lipid contacts. So this compares lipid contacts determined also by proton-proton mixing in NMR for intermediate 1 on top and fibril on the bottom. And to highlight here this section in this beta-2, beta-3 region, where in the intermediate we see contacts to the lipids, whereas with the fibril we did not. And so this also suggests that in the intermediate, these sections are in contact with some sort of lipid species. And in particular, these are the chemical shift of the acyl chain here, shown colored in red. Okay, so that brings me towards the end. And in summary, we've been able to identify and locate in the primary sequence a region of anti-parallel beta strands in an alpha-synuclein intermediate, which has connections to toxicity or possibly membrane disruption. And these anti-parallel beta strands, as I just showed on the last slide, have a lipid binding domain. Modeling of the oligomeric structure is something that we're continuing to do, and we're looking forward to extending the structural models to also include lipids and look at the stability of such structures. As Bob introduced at the beginning, we are also interested in interactions of a clinical drug candidate like ANLA138B with such oligomeric species, but I don't have anything to show you about that today for the oligomer. Acknowledgements are manifold. I pointed out Leif Entenschmidt already, who developed the procedure for isolating this intermediate species, and Brenda Sant. The external collaborators are both for cryo-electron microscopy, Gunnar Benedict, and here at the NPI NAT, Christian Dienemann. ND simulations are done by Bert de Groot here and Dirk Mathis. In-cell experiments, the toxicity experiments were done by Magdalene. And the photobleaching and cold data comes from NPI for Light. Transmission EM comes here from our facility run by Gudrun Heim. And of course, there are all the people involved from the NMR department, Christian Driesinger, who is intimately involved in all of these measurements on the fibrils on oligomeric species and is interested in the small molecule. And of course, the biochemistry team, Stefan Becker and Melanie Begstrode. So I'd be happy to take questions.
Okay, well, thank you, Loren, for a really nice presentation. I would start off with one question. So you talked about L1 and L2 initially, yeah, but you didn't say much about L2. Is that unstable compared to L1? And the other question is, how long can you keep L1 around? Is it infinitely stable, or does it convert to L2? Or
so I'll answer the first part, and maybe Brenda can answer the second, or I'll answer the second part. Maybe Brenda can continue. So the samples are stabilized by spinning them down and keeping them at about 4 or 5 degrees in the fridge, and they're fairly stable. And then in the NMR rotors, we can't go below about 20 degrees Celsius. At 20 degrees, they degrade or start converting over into fibrils within several weeks. Of course, it depends batch to batch a little bit how much time we get. So Brenda's constantly measuring reference spectrum and making sure that the sample is still what we want. And the intermediate too, we just haven't gotten to it yet. Brenda, do you want to add anything?
No, that sounds about right.
Yeah, well, I mean, so you could say C13 and 15, you could uniformly label the synuclein and continue. I mean, you seems like you've gotten a good start on doing the structure with the proton detected experiments, and if they are stable, you should be able to get a complete structure out of this, right? Is that
Yeah.
right?
Yeah, no, I know you're right. I mean, at the level of, I think, a basic dimeric unit, we already have a reasonably good structure. Difficulty is the number of different ways of then extending that to a tetramer. So, and what happens in the in-between region between the anti-parallel region and the parallel register.
Yeah.
So this is more subtle. I should also say carbon detect data we also have, but the proton detect is really nice because the yield of a oligomer is very low in this procedure. So we lose a lot of protein to monomer when we stop it at such an early phase of the aggregation. So what was the number? About 2% or so of the protein we use actually ends up in the rotor at the end.
Yeah.
And then, yeah, a further complication, which I forgot to mention, is that deuteration also impacts the kinetics. And so we weren't able to prepare deuterated samples.
Yeah, okay, so Rams has a question. Should we assume NMR sample to be 100% oligomers? You maybe sort of addressed some of this. How stable are the observed oligomers? Did you observe oligomers converting to form fibrils or monomers? Or did you have any changes to the sample conditions to stabilize the oligomers? This is in the chat if you want to try to read that.
Yeah, I don't know if I'll get all of that. But part of it, there's let me see if I can pull up a slide showing the conversion. So we do see some conversion in the rotor. So let me share it again, this slide. So here you can see some of the changes that occur. So Brenda, do you want to comment on that?
Yeah, so like Loren said, we record fingerprint spectra almost at a regular interval while we're recording some of these longer 3D spectra. And on the rightmost figure, you can see how this is pretty consistent over time. But there comes a time, depending on the sample handling, and it varies from sample to sample, that this spectrum eventually begins to change. And then you can see some very isolated peaks here. They begin to disappear. And this is when we know the sample has converted to the fibril.
Yeah, so we have some pretty isolated peaks that are characteristic, like these down here beyond 130 in nitrogen. Which helps with that. So
Bipin Agarwal has a question. Was a bit surprised by the homogeneity of oligomers, especially the whole ensemble transitioning from one intermediate to another. Could you comment on the lack of heterogeneity?
Well, I think we were all a bit surprised, happily surprised. On the other hand, it's a particular prep. So the PMCA or the sonication, which is occurring, probably has something to do with this. Of course, it means there must be some thermodynamic reason why the process is arrested at one point. So we get predominantly one after the other, rather than more of a smearing out between the different species. But this becomes very speculative.
Did you say what kind? Did you say the type of lipids that you're using? I remember.
I think it was on one of the slides, but I didn't comment. Probably it's it is also negatively charged. So we have PA and PC.
And are they 16 carbon chains or what?
It's PO series. So POPC, POPA.
Oh, okay. Yeah. Okay.
Yeah.
Yeah. So those are well above, yeah, a transition. Yeah. Okay. So I see in the chat, there seem to be some other questions. I'm not sure they're for Loren or for Emma. It says, thank you for the great presentation. For how long did you incubate the mixture of protein lipids and in which conditions? I think these may be for Emma. Right?
Yes, I think this one is for Emma.
Yeah.
Yeah. So for days if needed. I mean, so the I mean, if you have the proportions that make aggregation happen, that could happen within a couple of days. So that we recorded, and then of course those that were not aggregating within a couple of days were also sort of kept for these times. So it from everything image from early event and 24 hours or even later. So that was the question of the time. Was there something else?
Well, there's another one that says, great work. How do you envisage repacking of anti-parallel beta strands and the misfolding intermediate to parallel intermolecular beta sheet and amyloid?
But that is not for me, I think.
That's not
Yeah, that must be for me. So let me share this.
This is from
Sorry.
Olga Gorski.
Yeah, let me share the screen one more time. This is a very slow movie showing a dimer species transitioning from anti-parallel to parallel. So I could not figure out how to speed up this movie, but you can see it. It is, at least for the dimer, something which can be imagined.
Yeah. So, Loren, another question I was going to ask is, how similar is your model now to, say, Chad Reenstra's original Greek key alpha-synuclein structure? Have you? I mean, there are so many alpha-synuclein structures these days.
Yeah, there are more and more, and so I think this is also touches on a more general topic, which is which of these are relevant. Since there's more and more being found every day, you know, who knows if this will be found to be relevant in a particular biological context. It's all in vitro for now, and the Greek key structure is really quite different from the L2 structure. So if you look at the position where there are strong kinks between beta sheets, these are different, and the whole structure is quite different.
So does your fibril then convert to the Greek key structure, or does it?
No.
Converge? It wasn't converged.
No, I think the last thing that happens is so the second to last thing that happens is some of the C-terminal part of the sequence forms into the fibril, and then the final thing that may happen is some of the very N-terminal residues forming fibril. This is seen in the cryo-EM more so than the NMR. So residues 1 to 14 or so, then also can form part of the fibril. But once that fibril species is formed, it's very stable.
Yeah.
Yeah.
Okay, I don't see any more questions.
There is one more question for Loren, which is not in the Q&A, but in the discussion.
Oh, sorry.
Yeah, it's okay, Emma. It's from Emma. She has a question about the fibril structure with associate micelles that you show in the beginning. In the cartoon, in the paper, micelles seems to be located in the middle of the fibrils between the filaments. How did you come to this conclusion of micelle location? Sorry.
I'm not entirely sure I follow the question, but so
Emma, you can ask
the positions of the micelles in the MD simulation, this is just what's seen, but of course they're moving up and down on the filament, And in the cryo-EM reconstruction, then these are kind of smeared out. So they.
But in the paper, there are mices apparently that appear to be located sort of in between the filaments within the fibrils. And is that I mean, it's clear that how you come to the conclusions from micelles and so on, but I wonder, is there data? Well, what is it that makes you draw the conclusions that are sort of located in the middle and not on the surface, or are they on both?
They're all also on the surface. So
do you know that they are in the middle?
Yeah, so I didn't show it, but just like with the oligomer, we also have NMR data showing contact to lipids, which covers some of the same regions. This becomes a little bit tricky though, because in cryo-EM, there are 6 different species by NMR. We measure only 2 or 3 of those, depending on which conditions. So we optimize conditions to get L2, the basic filament, but we still have different overall arrangements. So filament-filament interactions are different. And by NMR, those are all averaged. But we do see contacts to lipids for the regions that are expected. So both from the densities, first of all, but then also from the NMR, we can then point to that being lipid. There really isn't much else in the sample. So these are in vitro preparations. So for extra densities, they don't look like protein. So it's either protein or lipid.
Yeah, I understand. I was just a bit surprised about that location.
Yeah, so I would say we were also very surprised. In fact, some of our earlier attempts to do the cryo-EM involved discussions about how to get rid of the lipids. But in the end, it turned out that actually visualizing the lipids as part of the structure was some of the most interesting part of the work, I think.
So Loren, it's a great talk, great data, great stuff. How did you said only 2% population is oligomer, lipid bound. That means you got tons of monomers and maybe a little bit fibers as well. So what exactly did you do to isolate your oligomers?
So at that point, the sample is spun down together with lipids, we get enrichment then of both lipids. We have a lot of lipids packed in the rotor, and then also oligomers.
So I say monomer is supposed to be bound to lipids as a helix, right? So when you spin it down, that should also sediment.
Brenda, do you want to comment more? So we tried both lipid protein ratios of 5:1 and 10:1.
And I mean, so there are, if you look at the spectrum of a lipid-bound monomer, there are some salient peaks that belong only to that spectrum that we don't now see in the intermediate 1 spectrum. So of course, there's a possibility that these species exist below the noise floor, but at least they don't show up in the spectrum.
So do you think the monomers are weakly bound as a helix, don't survive the spinning condition maybe?
Or maybe by now, if there's really this homogeneous transitioning, maybe all of the lipid-bound monomers are actually oligomers. They have now converted to oligomers. Yeah.
Okay, thank you.